how does it, because that could provide a developmental link, right?
Oh, I see.
If it was not directly feeding into toll.
Yeah.
Well, we can talk about.
I don't think we know, but I think we should find out.
We're in Drosophila.
Yeah.
Which is the only thing I know. I mean, you use the toll pathway for generating the dorsoventral polarity.
Yep.
As well as for innate immune response.
Well, here I'll say this because I think it's interesting. One of those host CD-NTases here is actually fundamentally involved in ocular tissue development in humans and in mice. So it's a gene called MAB21L1. And the only reason I say that is because CGAS is called MAB21D1. So these two enzymes are really similar. Humans with mutations in MAB21L1 fail to develop their ocular tissue. It's a very severe congenital condition. And I think that says that there's a cyclic dinucleotide made by that enzyme that goes on to control tissue development. And so hopefully, what we're going to find is some of the bacteria are manipulating tissue development in some way.
Are enzymes known that break down these molecules?
Oh, gosh. I can't wait to have some time to get back in the lab and find out. We've got great radio-labeled cyclic dinucleotides that we can simply incubate with lysate from a liver homogenate, from a eukaryote, or from different bacterial homogenates. What I'll say is we don't know of any phosphodiesterases that are dedicated to these novel molecules, but these molecules aren't typically broken down by conventional RNases. Something about that constrained conformation of the cyclic dinucleotide means they typically have dedicated phosphodiesterases. So it'll be fun to find those.
So you mentioned that these CGAS-like enzymes are included in vertebrates, right?
Yes.
So, I mean, were they, do other eukaryotes lost it, or was it acquired by?
I should say, actually, all metazoans have CGAS-like enzymes. We know the most about CGAS, the DNA sensor, in vertebrates because all vertebrates have the DNA binding capability of CGAS to interact with that DNA agonist. If you go beyond, say, to arthropods, which have many CGAS-like enzymes, they've lost the ability to interact with double-stranded DNA. And so there, I don't think we know what those do, but you can go to a sea anemone and there's, over 10 different CD-NTases. So there is a group of people in the lab who are working on reconstructing the eukaryotic development of these CGAS-like enzymes, and so that'll be cool to find out what's going on outside of the metazoan, or outside of the vertebrates.
So dinucleotides are easy to form, but I mean, trinucleotides, it seems like you'll need a translocation mechanism. And so, I mean, are there two active sites, or is there just a translocation mechanism that moves the?
This is what we're trying to figure out. So CGAS-like enzymes being polymerases means that they have one active site and they form bonds sequentially. So the way that they make a cyclic dinucleotide is to start with ATP and GTP, make a linear intermediate, disassociate that intermediate, it flips around, reassociates, and then that cyclizes the compound. And there's a lot of argument over, is it real dissociation, is it just what exactly is happening? But we understand how to make two bonds and have an active site that can accommodate two conformations. But the cyclic trinucleotide synthase implies that it now has to be 50% better. It has to have a third conformation. So I've been trying to get crystal structures of CD-NTase38 in different conformations with different reactants, and we're having a hard time trapping the intermediates, but I think it's going to be cool. But we have no evidence for a second active site.
But are you making the assumption that it comes out, flips around, and then goes back?
Yeah, I don't think it's processive. So there are CGAS-like enzymes in the host, namely one that's called OAS, that processively makes an oligo A. And that will make between 2 and 10 adenosines that are linked processively. But that's because OAS lacks a structural feature that sort of condenses the active site and controls it. CD-NTase38, I think, can't function like OAS, but it's certainly, it's not outside the realm of possibility that it's
But the kinetic
rotating.
profile should tell you if it's coming out or going back in, or if it's processive.
Yeah.
Is it distributor or processive? Careful, kinetics should give you a hint of that.
And that's exactly what I want to do going forward.
All right, last question.
Yeah, so even though the cyclic trinucleotide doesn't bind to STING, does it still activate the type 1 interferon response?
So what I want to do with all these nucleotides is introduce them into cells and profile what each one of them is capable of signaling. So not just look at type 1 interferon, but look more broadly. I haven't introduced the cyclic trinucleotide and looked at type 1 interferon yet. But that'll be a great thing to do because hopefully it turns on an orthogonal type 1 interferon pathway. That would be the dream.
Right. And then what about like an alternative to Recon? So Recon is only in mouse, but there's no human homolog for that. Any thoughts on that?
That's, you've read your literature. Yeah, so one of the funny things about Recon is that it's an alloketo reductase that's only, we haven't been able to identify a homolog in humans that can bind cyclic dinucleotides. But Recon is part of a family of 13 other alloketo reductases, all of which can participate in that NF-kappa B signaling pathway. So I think a great targeted and candidate-based approach is to simply ask, what do each of those Recon homologs bind? Maybe they each have different specificity. And I think going into the human Recons is probably a great place to start, right? So that we don't end up finding a mouse protein that works great and missing the human aspect. But I think it's a matter of looking at these other alloketo reductase homologs.
Thank you, Aaron. That was
Thank you so
excellent.
much.